thing that I hope you're getting pretty good at is realizing that the way that the nervous system and the endocrine system work, there's a lot of crossover there. Sometimes I say that's gray area or a gray zone because ligands are not generally isolated to one of those two systems. They can be in both systems. And I do love the example of how the beta cell in the pancreas works because it brings together this idea that in human physiology in the human body, there is a lot of repetition in the methods that we see in the physiology. The beta cell in the pancreas is an endocrine cell because it makes insulin, it secretes insulin. You've already learned that. But what you might not know is how the insulin gets out of the beta cell in the pancreas. And this is the interesting thing. The beta cell in the pancreas is electrical. It's an electrical or electrically excitable. And that is strange because we are accustomed to seeing only neurons and muscles being electrically excitable. This means that they work by sending charged particles across their membrane. So here we're seeing that same kind of method being used in an endocrine cell. Let me show you how it works. So inside the beta cell in the pancreas, there are vesicles. And you know already that these vesicles are storing insulin. Insulin is polar, so it can be stored inside of a membrane-bound vesicle. That's what it looks like. And the other thing you need to know that you can bring over from the nervous system, or you'll be seeing it again in muscles, electrically excitable means that it has a resting membrane potential. You've learned so much about this already, hopefully. In order to get this vesicle to fuse with the cell membrane, it needs to have exocytosis. So we're going to be looking at electrical excitability in the cell, opening of voltage-gated calcium channels eventually, and making that calcium trigger the exocytosis fusing of the vesicle. So let's look at how this happens. The first thing that you need to know is all electrically excitable cells have a resting membrane potential. So that would be step one. In other words, nothing is happening to this pancreatic beta cell. It's resting, and it's negative inside compared to outside due to leaky potassium channels. And you already know that because it looks like a neuron. It looks kind of like a neuron where potassium is leaking from high to low outside the cell. Then what happens is we have a situation out here where all of a sudden the bloodstream has high blood sugar. Now, living inside the membrane, the transmembrane reporter GLUT transporter lives there at all times, and so it can take the glucose from high to low into this pancreatic beta cell. Now, if you remember that all cells prefer glucose, you can remember what they do with that glucose. They start breaking the bonds and getting it into a form of energy that the cell can use to make ATP. So all cells do this. That's why we say glucose is the preferred energy source. So when the glucose gets inside here, it gets broken down by glycolysis, citric acid cycle, electron transport chain. That's my abbreviation. So step 3 would be, sorry, I should number these for you. Step 3 would be we're turning that glucose into the energy of ATP. And that you've already learned also. So when ATP is made here, ATP is going to have a new function that you haven't seen yet. This is actually the only new thing that we're adding to so many existing mechanisms that you already know. And this new thing is that ATP is going to be used as a ligand. That is weird, but we have seen this happen with other things, with other what we call second messengers, where they appear on the inside of the cell, and it can come over here and it can actually close this potassium channel. So ATP actually comes over here, closes it. It's not acting as energy, it's acting as a ligand. So that's the new piece of information. It's used as a ligand to close the leaky potassium channel. You already learned that leaky channels can be closed from the inside, and the new piece of information is we're using ATP to do that. When that happens, everything else is pretty easy. You know that that will depolarize the cell because it's keeping a positive inside. Okay, so when the positive can't leak out, it stays inside. That's step 4, depolarization. And then depolarization leads to the voltage-gated calcium channel opening. And then the calcium will come in from high to low. That's step 5. And finally, step 6 will be exocytosis. And that is how we get the membranes to fuse, where the vesicle becomes a part of the bigger membrane, exocytosis, and the insulin will exit the cell. So didn't quite get all the numbers on there, but hopefully you know that when it closes the leaky channel over here, then that will cause depolarization, and that's step 4, because potassium stays in. Step 5, the calcium channel, and step 6 is going to be exocytosis. This is in a weird place in your book, but I love it. So you might want to look that up. Chapter 5, it just shows you the beta cell at rest. It's weird because it's electrically excitable, and it is not sending an electrical message here. So the leaky channels are open, voltage-gated calcium channels are closed, insulin is not being secreted. However, when we have this trigger in the fact that the blood sugar is going to be increased outside of this beta cell, so if I can get that better for you, then the glucose, or sugar from the blood, moves over and ATP is made, closing the potassium channel, and then calcium voltage-gated channels will open upon depolarization. Just a great example, really, of how you can bring together so many things that you know already and look at this in the human body as, systems can have repeated methods, okay, an electrically excitable endocrine cell. That's what we're seeing there. Okay, so back to our primary focus of this nutshell. I want you to remember what negative feedback is. Negative feedback we often see in the endocrine system where we have a stimulus like high blood sugar, and then the response in the body will happen and remove that stimulus. Okay, so when all of these things take place, insulin is out in the bloodstream, it causes the blood sugar and over here to decrease, and then there's no longer a stimulus here to the beta cell in the pancreas. So that is negative feedback. You need to remember that definition as we go into the hypothalamus-pituitary pathway. So let me back up here and I'll write it down for you. Hypothalamus in the brain comes down like this, and it has this stalk hanging off of it, which is the pituitary gland. In this example, we're going to be talking about the anterior pituitary only, okay? Later on in the semester, we'll talk about the posterior pituitary. But for today's negative feedback loops, I'm just doing the anterior pituitary. And I would like to focus on the pathways that you'll be doing in your lab, also for the application questions in your book problems. So what you need to know is the hypothalamus has neurons. And again, here we go with systems using repeated methods. It makes a chemical messenger which gets dumped into the bloodstream. So now it's a neurohormone. The first pathway I'd like to talk with you about, we would imagine that this chemical or neurohormone is TRH, made in the hypothalamus. It travels through this little portal system, which is a small set of capillaries, right over here to your anterior pituitary. So it's not going in what I call the big body yet. It's just traveling over here to the anterior pituitary. Okay, so the anterior pituitary has endocrine cells. Now, they do make hormones, and they can be stored in vesicles. So you know that they're polar. So here's the hormones being stored there. However, it needs to be able to respond to this trophic hormone. A trophic hormone, by definition, means it causes the release of another hormone. So TRH is a trophic hormone. So it's going to have a receptor for TRH in the endocrine cell here. And when TRH binds there, this hormone is going to be released into the bloodstream as well. It is called TSH, thyroid stimulating hormone. The way I remember that is R comes before S in the alphabet, and that really helps me. Okay, so the TSH goes into the bloodstream, and now it is also a trophic hormone. It is going into the big body to find its endocrine gland so it can cause the release of another hormone. So TSH is a trophic hormone. Now, it's going to go to the thyroid gland and cause the release of the thyroid hormones. So down here in the big body, where your thyroid gland is, it has the TSH receptor. So it can respond to the TSH trophic hormone, causing the release of thyroid hormones. So we have 3 hormones in this pathway, and then thyroid hormones will go into what I call the big body, and they will go to many, many cells. Yes. So T3 and T4 in your lab, it is called thyroxine. And so I'm going to call it thyroxine. And it's going to go to many cells. Almost all the cells in the human body have receptors inside of them. Remember, this acts nonpolar. And so to many body cells to increase metabolism. And that is what you need to know for your lab. The other place where TSH goes, in addition to the thyroid gland, is it travels back up to the hypothalamus, telling the neuron that makes TRH to stop doing that. That is called negative feedback. It feeds back up here and turns off its stimulator. Thyroxine in the same way can feed back up here, turning off its stimulator, TSH. It can also come up here and turn off TRH. So that's the negative feedback, where the hormone further down in the pathway will turn off its stimulator. We could also do this for the cortisone pathway, and that's an example that you need to know. Coming from the hypothalamus, we would have CRH. Goes down to the anterior pituitary, and there will be an endocrine cell waiting there, and it will release ACTH. ACTH is a trophic hormone also. It travels in the big body to the adrenal cortex, and that is where you will get the third hormone release, which is cortisol. It also goes up here to turn off its stimulator, CRH. So ACTH is going 2 places from the anterior pituitary. It's going up to turn off its stimulator, CRH, and it's going down to the adrenal cortex where it causes the release of cortisone. And again, cortisone will turn off its stimulators with negative feedback. This is really important because if you have a patient who has high levels of cortisone, it is not necessarily a pathological problem with the adrenal cortex. In other words, that patient could have a brain problem here or here causing a high secretion of cortisone. So we need to look at the pathology and see if there's high cortisone levels. Is the problem in the adrenal cortex or is it in the brain, two regions of the brain? Of course, if it's in the adrenal gland, that's an easier problem to fix. Same thing over here. If you have high levels of thyroxine, T3, T4, it could be a problem in the thyroid gland or in the pituitary gland or in the hypothalamus. So you need to be able to pinpoint the problem before you go into the human body and try to fix it. Okay, so good luck with your online labs. If it has to do with this pathway, let me know if you have questions. Thanks.